Hello. Welcome. Oh, how's everyone doing? How was the weekend? Looking at the number of folks in the Zoom room, I'm gonna guess that the weekend was pretty busy. We hope you're safe. We hope you're okay. If you're sick, stay home. Yeah, all of those things. If you have any questions, pop them in the chat. We love to talk about things that we have opinions about.
We had a good weekend.
We had a great weekend.
We had, I had to host in the Zoom environment. I hosted a conference with people from all over the country, and which was lovely. Yeah. And I walked almost 50 kilometers during it because I have a treadmill desk, and that's pretty lucky, and I love it. And then I used 13 cups of flour on Sunday. I made a pie. I baked bread.
Yeah.
I made muffins.
Yeah.
I made pancakes.
Yeah.
I made crepes.
Very good.
Yeah.
Very good. They were really, really good pancakes. And I cooked with how many pounds of tomatoes and squash?
You reduced a lot of the garden into delicious chili format.
Food. Yeah. So it's a garden harvesting weekend. Okay.
A Zoom session.
Yeah.
Did we all go to home? Well, homecoming in that we're home.
We're home and we stayed here. Yeah. And we didn't go to homecoming.
It's an awfully specific amount of cups of flour because it was a recipe. It was like 6 recipes. 5 recipes?
I feel like if we had gone to Chancellor's Way, we probably would have been like the people going, whoa, 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 everyone. Oh, man.
You're going to stain the couch. It's going to get stained.
What happened to the couch? Somebody let us know.
Fire? Yeah.
Was it the couch that was on fire? We have questions.
Yeah.
Okay, good. Let's move on.
Oh, I'm so sorry. What should we do if our roomies are sick but we're fine?
If your roomies are sick but you are fine, wear a mask all the time. Yep. N95. If you need one, let us know. And what else should you do? You should open the windows all the time. Good ventilation is really important. You should make sure your roommates flush the toilet with the lid closed so that spray doesn't get around.
And that's actually a real thing, the vortex.
Yeah. You should obviously wash your hands, wash everything as frequently as possible. And if you can build a Corsi Rosenthal box, we'll pop a video in the chat on how to do that. Those things can be really important if you have poor filtration in your home.
This is true if you're in residence or all sorts of things. There's a couple of quick, but if you Google Corsi Rosenthal.
Yeah. Okay, more questions, please. Like, this stuff is important. Pop it in the chat.
And if I miss it, because it doesn't seem we're responding,
we will get to you. Pop it in again. These things are important. Cool. So let's try to focus on tree reading.
Y'all have toilet lids? It's like, we're fancy schmancy.
Right. We're part of the privileged group that have toilet lids. Yeah. If you don't have toilet lids, man. Sorry about that. Okay.
So there's several questions about where to meet in the dairy bush. I believe in the instructions, there's a video there where you follow an old course coordinator out, but it's still the same place. It's basically walk straight across across the crosswalk, head towards past the gray barn, past the red barn, continue straight. When you get to that forest at the base of the hill, just stop there. That's the dairy bush, that forest.
Have a wonderful time there. Rain or shine, if you're going to go. It'll be great.
Get the full dairy bush experience.
Yeah, breathe some clean air, enjoy, listen to the traffic in the background. Okay, shall we?
Past the cows. Yes, it does say past the cows, but I think it's past the cows.
Past. Okay,
The video in the email doesn't show up. I can see that potentially being a problem. It is on your...
It is on the announcements page.
The announcements page.
Yeah.
It's embedded there as a video.
Yeah. Okay. Amazing. Are you ready now?
Sick today, have the dairy bush. I can just join online, correct?
Get in touch with the course coordinators
if
there's like a last minute change and all of that at the course email address. Okay. Now can we start? Are we good?
I think so.
Yeah. Okay.
There was some tree damage. That's sad.
Oh.
And yes, it was the couch that was on fire.
It was.
Yeah.
Holy shit.
Res bathrooms have no lid. Oh my god.
Oh my god.
This reminds me of 1070 in res. Remember in February?
We will take to Twitter and we will ask for lids because gross. Okay, here we go.
Oh my god. I'm sorry when I laughed. I thought that was just
Yeah. Okay.
Douglas.
Sorry, Doug. Okay, here we go. Let's get started. Thank you so much for drawing. It totally warms our hearts. And we're going to clear everything for now. There we go. So tree thinking is super fun, a totally kind of nice way of organizing information and being able to infer things, which is also super fun. So we're going to do sort of some of the basics just to review to make sure that you've got it. But remember, as we move towards the topic test, we want you to sort of become sort of next level in terms of tree reading. So we'll introduce a couple of like questions. Some of them will be familiar. We'll take it up just in case there are some misunderstandings. Really cool to focus on what the phylogenies actually say versus what they don't. And you'll see what we mean. So we don't want you to make any assumptions. So what we're going to do is we're going to kind of bring you to the edge of making assumptions, and then we're going to kind of dial you back. Okay? Does that make sense?
Look at them.
We'll look at them.
Look at those assumptions.
Okay, but first, Smith is going to rant a little bit.
Yeah, so this question comes up a lot. In fact, you can look at it in a pictorial format. Are we descended from monkeys? And if so, why are there still monkeys? Maybe you've run into this before in debates or in your training as a biologist to this point. But if you Google kind of phylogeny or evolution in the Google space, often as an image search, things like this come up. Now, some of these you're going to we'll actually talk about later on. Like I see nested in there a lovely picture of some Darwin's finches, and Darwin's finches is an amazing example of evolution via natural selection. But one of the things that pops up consistently here is this really harmful view, limiting view of evolution as a progression, that you started from a little person and you became a medium-sized person, and then you get a spear, and then you're like Jason Momoa, and that's like somehow the progression of evolution, and it's wrong for a couple of things. And one of the principal reasons is that it's wrong because it's linear, and the other is the principal reason that it's wrong is because it's a progression. And we're going to try and unpack some of those miscalibrations of evolution today, and we're going to do it with tree-based thinking.
Right. So what we're going to do is we're going to kind of review some of the things from online. And we're not going to review it, actually. We're going to sort of see where you're at. Okay, so it's going to be a lot of stamping, and we're going to ask you to just follow the instructions again for when to stamp and all of that stuff. You guys are so amazing, and we're just so grateful at how good you are at this so that we can do this together. So what we're going to do is we're going to pretend that we have 5 modern species. 5 species. Right. And we're going to represent them with different colors. Okay, so we have black and green and red and yellow and blue. Cool. And we're going to start looking at a tree, and we're going to make some statements, and we want you to get your stamps ready, but we want you to just wait for that magic number 3 for us to stamp. And you can test your stamps anywhere that's not like on the white space. That's great. Okay, cool. So here we go. Ready? Oh, Nina needs to come in. Oh, and Carly. Oh. Welcome, welcome. Yay, look at the testing. Okay, here we go. So we'll ask you to hold off on stamping. Here's our first statement. So remember, I'm going to say that magic number, and that's when I want you to stamp. So yellow is the outgroup of the other 4. So if yellow is the outgroup, where on the tree do you stamp? In 1, 2, 3.
Oh. Okay. That's awesome.
So we're good with outgroups then. We're good at identifying where the outgroup is what I'm getting from this. And that's why I want you to hold off till 3, just in case there are some folks that are unclear, because then we'll stop and explain. Okay. But so far it looks good. Okay. Super. Excellent. Okay. So I'm going to clear now, and we will continue with the next statement, okay, as we build this phylogeny. Okay, the next statement, again, please hold off and just prepare your stamps. Oh, right, we'll just take this out. Yellow is the outgroup. So this is what it's going to look like, okay, right? And then we have these other 4, and they are unresolved, right? We're going to resolve this whole tree in a second, okay? So blue and red are sister taxa. So if that is true, in 1, 2, 3, show me where the red is. Okay, good. We have some disagreement. That's helpful. Thank you.
Very helpful.
Very, very helpful. Okay, so we're going to hold off for a second. And I'm going to delete everything here. So sister taxa, let's try again. Okay, don't stamp yet because we have some disagreement. So hold off. I'm going to clear.
Hands off the stamp.
Hands off the stamp. So what if I said a sister taxon is the one that is most closely related to blue? And 1, 2, 3, go. Still some. Okay, good. So you've moved it over in the right direction. Yes. So most closely related to blue means that they have most recently branched off. They have a more recent common ancestor. So that's good. If you have any questions about that, if you were stamping a little bit further down for a less recent common ancestor, pop them in the chat. Smith will clear them up. Okay, good. Now we're going to clear again. I hope this stuff is helpful.
Be safe, y'all, with the fire alarm. Listen, leave, come back. We'll still be here.
Alarm. We're still recording.
Or you're going to catch us on the YouTubes.
Yeah, okay. Next. Oh, wait, we're going to just show you this. So red and blue are sister taxa. So now we've resolved the relationship between red and blue, and we've resolved the relationship of yellow to all of them, but we haven't resolved green and black. We still don't know the order of the branching here, okay? So let's try another statement. And again, just hold off a little bit because this one is a more tricky one. Green is more closely related to red than black is. And what I'd like you to do is to show me in 1, 2, 3 where green is. Good. Excellent.
That's great.
Excellent. Okay, you're doing really, really well. That's great. Okay. Super. Any questions about that? Pop them in the chat. Well done. Okay, so that's kind of basic, like, right? Tree reading 101. And you've got it, which is great. So I'm going to clear, and we're going to move into resolving this. So green is more closely related to red than black is. Here's what it looks like. Here's what our tree looks like, right? So the idea is that the more recent the common ancestor, the more closely related. And this is important. So let us we'll drill in on some of the assumptions and some of the ways that you can make sure that you understand a tree, okay?
So this is a really powerful way of thinking about,
really cool.
about evolution. There's a bunch of, we talked with that image of the screen capture of the Google image search about 2 different misconceptions that people generally have of evolution, that it's linear and that it's progressive. And tree-based thinking goes a long way to helping resolve that, that misconception. And we'll talk about some of the literal twists that we can do in the branches.
Okay, so one thing that's important to note is that you can draw trees in many different ways. So at the beginning of this unit, we actually showed you a circular phylogeny, right? That was more like a circle. Here are 2 other representations, the more like football goal, and then this slanty one. Which one do you prefer?
I always like drawing the
slanty one?
No, the rectangular.
You like the rectangle?
I do not. If I'm just drawing it, like if I was going to pull up this notepad and draw that tree and show it to the camera, I would start out with the rectangles.
With the rectangles. Yeah. Okay. Good. Okay.
This is a great question. What if red didn't exist?
Oh,
would blue and green be considered sister taxa?
Yes.
Yeah, that's a great question, and yes.
Yes. Excellent. Well done. Okay.
That's actually a great way of answering it with an example, an earlier question. Are the sister taxa the ones touching? Or the yes?
Yes.
Yeah.
Okay. Folks still coming in. I guess the fire alarm is over, maybe? I hope.
Yeah.
Okay, here we go. Continue.
We found a new spot.
Yeah. Okay, now, there's something that's super important to note. This is it. Do you want to explain?
I love the fact that you can no, you start.
Okay.
Yeah, and I'll.
So the thing that's really important, there is, when we look at graphs and when we look at figures, it's really important to recognize what parts of those things have no information and what parts do have information. In a phylogeny, there is no information in the order along the top. Okay? It doesn't matter. And so we can represent this by actually just twisting the nodes and showing that the relationships are the same. Okay, so there is no information in where yellow is along the top relative to green. So you can see that we've just twisted the nodes, rotated them, and now yellow is beside green, but the branching is the same, right? And that's the important part. So it's the branching from top to bottom or bottom to top that tells us about the relationships. There is no information in whether blue is on the left or on the right of red. That they have this more recent common ancestor is the important information. Okay, is that good?
Yeah.
Okay, good. I hope that was clear. So basically you can't read across the tips, okay? Can't go this way, and people do it all the time. Yeah.
Yeah, and in fact, that last, like that blue, if you go back just for a second, this one in the upper left-hand corner, that kind of phylogeny that we resolved and you colored, people often will read that, and if you were to put people on it, we'd be like, well, we're over there on the right-hand side. We're like the ultimate expression of evolution. It's like, no. And we'll come back to that.
Yeah. Okay. Cool. So, Clade, you're up.
Yeah. So we've got some new terms to revisit just together. You've read about them in the pre-lecture stuff. And these are important words, and they're actually derived from tree-based thinking as well. So clade is a group of organisms that consists of the common ancestor and all of its descendants. So clade is synonymous with another term, a slightly longer one, called monophyletic. And so the clade or a monophyletic group has to include that common ancestor, and it has to include all of the descendants. And if it doesn't have one of those things, then in one of two ways, it's not monophyletic. So the most important thing for you, and we'll show you some examples of these non-monophyletic groups, they can all either be poly or paraphyletic. Paraphyletic is because it's missing at least one descendant or the common ancestor. And polyphyletic, this example here, is one jumping across, or maybe we ask about that question in a second, I don't remember. But I want to just stress, the important thing for you as a biologist is to be able to identify monophyly. It's less important to be able to say it's polyphyletic or paraphyletic, because in fact, determining those two things really has to do with the information that you've got in front of you. And since these phylogenies, every one of them is just a hypothesis, it might not be correct. So be able to identify monophyletic groups, the one that includes the ancestor and all of the descendants.
That's right. And for the purposes of 1070, we're just going to use the terms monophyletic and paraphyletic on the topic tests, okay? So if it's not monophyletic, let's just call it paraphyletic for now. If you take invertebrate or the invertebrate biology, then we'll bump it up and add some more detail to that in a second year course, okay? Good, thanks. So basically, and we don't want you to overthink this, because we do find that a lot of folks really try to overthink this whole monophyletic paraphyletic thing. The idea though is two things are important. The existence of a common ancestor, and the idea is that, yeah, like we all have a common ancestor. So there is a common ancestor when we put together, relationships. But then the next question you want to ask is, does the grouping, once we've established where the common ancestor is, does the grouping then include all of the descendants, okay? And that can be a little bit more complex because oftentimes we talk about birds and mammals, right? We talk about them with respect to conservation and all of these things, right? So we talk about birds and mammals. Great. Could you please give me a stamp on where the common ancestor of birds and mammals is? Excellent. Okay, right down here. Good. So now that we've established where the common ancestor is, show me then which descendants would need to be included for this to be a monophyletic grouping. Yeah. Excellent. You got it. Okay. So then birds and mammals, if we just look at that grouping of organisms, is a paraphyletic group, right? Because if we wanted it to be monophyletic, we would have to include all of these other descendants. So well done. That's great. You okay with that? Any corrections you want to make?
Yeah, paraphyletic, because when we talk about reptiles, we miss birds. Yeah.
That's right.
Yeah.
Awesome. Well done. Okay. So, which, hold off, no stamping.
No stamping.
No stamping.
Lift your hands.
Which of these groups then, A, B, C, D, or E, is not a clade? Hold on, I will send you a poll.
So we've got 2/3 of you who have voted. And
Okay.
remember, the important thing with these quizzes is, or the polls, is that is not necessarily that you get the right answer. It's that you decide what your commit to your decision. And then if it's wrong, that's actually that's the most fertile place to be learning.
Okay, cool. And we'll just give you a few more seconds. Good. Let's get it up to at least 80%. Go, 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 go. Maybe not.
Two more. One more. One
One
more person.
more. Just click.
Just vote.
There! Good. Okay. Excellent. We're going to end it and share results. So the correct answer is yellow and black. So well done. If we just have a group like birds and mammals, right? If we just have a grouping of yellow and black, the common ancestor is down here. And then we want to look to see if the grouping includes all of the descendants from that common ancestor. And of course, it doesn't, because it would have to include the entire clade or the entire group.
And I think this is important to bring up a couple of things here. One of them is that all of these poll questions are really ways for you to practice for the topic test, right?
Yeah.
Because when we write the poll questions, we are writing topic test questions. And so this is one that you read carefully, and it's not a clade. So that's kind of one. That's the pragmatic thing, the operational thing. The content-based thing is to recognize that phylogenies, each phylogeny has many clades.
Yeah.
There is not just one of them in there. There are many of them, and it's really context dependent. It's where are you drawing? What groupings are you referring to on the tree?
Yeah. So, question for you, if you want to pop the answer in the chat, how many clades are represented here? Cool.
I like it.
Yeah, that's good. That's good. Okay, at least 4. There could be more, but at least 4. Very good. I'm going to clear.
Clear away.
Okay. Okay. So it's really important to also note what units we are using when we talk about different phylogenies. You can do a phylogeny of mutations, and we've been seeing a lot of those lately in the news. So COVID-19 obviously is not a different species, but it's got a series of mutations, and we can show the different mutation events on a tree. More commonly and more practically within what we're talking about with 1070, as well as when we're talking about evolution more generally about, across species, we use those nodes or those branching points to represent a speciation event. So a place where basically there was this barrier to gene flow in a population that allowed for independent evolution that eventually led to a speciation event where at least one population or subpopulation became or evolved to be another species. Now, there are all sorts of nuances and delightful things that can happen, but this is generally how we represent them, right? Can a population be divided into 3 and you get 3 independent species? Yes. Can one of the species sort of remain and then the other one kind of branches? Yes. There's all sorts of these nuances, which is delightful if you're interested in studying evolution. It gets lovely and complex. But let's kind of keep it nice and simple. So the idea is that there are speciation events that happen at these nodes. But it's also important to know then what's being represented at the tips of these branches. And so you have to know whether it's a series of different species or whether we're talking about a mutation tree that shows these different mutation events. So that's one thing that you want to look at when you're looking for to understand what a phylogeny is.
So just like you would in graphically presenting things, like you'd label your axes,
yeah.
The units of change on a phylogeny should be easy to find.
Yeah, exactly. Thank you. And we'll show you some mutation trees as well. But first, let us ask you a question.
Let's check our ability to apply these concepts.
Very good. Tree reading question. Here we go.
And everyone's back from the fire alarm.
Good.
Or coming back. Remember, if your poll, some people had mentioned that their polls are not working.
No
Fine.
worries.
Put it in the chat. Declare to yourself. Write it on your hand.
Yeah. It's fine. Just commit to an answer.
Shout it. Like Michael Scott declaring bankruptcy.
In the middle of the library. Go, the
answer
is...
right! I declare.
I never know the answer to this one. Let's go. Okay, we're looking for a false statement.
Xavier really wants in. We are 2/3. Let's see if we can get to that 80%. Yep. Is there a way to watch these lectures virtually? Yes. YouTube and the content section under lectures, under content lectures, under lectures, lecture 5, there will be a link to the PDF of these slides with all of the links within them, and also to the YouTubes where you can watch at 1.5 times speed.
Okay, we're going to stop it. 70% have clicked in. We'll just stop it in about 10 seconds. Go ahead and see if you can click.
When does it get posted? Yep, it usually takes me an hour or so to get it up there. But it'll be certainly the same day and often within by the end of your next class kind of thing.
Okay, we're going to end it. Here we go.
Oh, we didn't get 80.
We didn't get 80.
I am sad.
Aw, I am sad. Share results. So here we go. The results. So the correct answer is E. That is the false statement. And if you said otherwise, that just should be a flag that you need to go back and take a look at some of the assumptions and some of the things that are related to tree thinking. And if you have a question, pop it in the chat or stick around after class.
Some of the assumptions and some of the powers of tree expressing things with a tree. And the one here being, of course, that when you rotate at the nodes, you're really not changing the information.
Yep.
Order across the top tells you
nothing. Okay, good. So let's go back. Here, so just a few little sort of added bits of information that can be really helpful.
And this one actually has already come up in the chat. Somebody was asking about this, why we could possibly, when asked about how many clades were in the last phylogeny, we said 4 at least.
At least.
And it was like, whoa, whoa, whoa. What do you mean at least? And this is what we're referring to. So it's a representation of the phylogeny. It's a simplification. It is a hypothesis. But within each one of those things, there's each one of those branches, there's many, many, many increments of time changes that are simplified. So just because it's pictured as one doesn't mean that's kind of simplified to tell you the message, to tell you the story. Within it, it's like Google Earth. You zoom in, right. Zoom in, and you can see more of these changes. And we've shown this one here, how it would look if there was a tree that had sea cucumbers, crinoids, urchins, and feather stars. I guess sea stars. And us.
And us.
Which would be a responsible phylogeny, because we're...
Somebody's like, me.
We're all deuterostomes. Today, this is... Oh, could you put a circle around the sea cucumber?
Yeah, this is us.
That's me. Until coffee, it's the sea urchin.
Prickly bill. You're lovely in the morning. Okay. The other bit of information that we're hoping to clarify here for you is the idea that the axis, and it's not always the y-axis. It depends, right? For the direction of the phylogeny. But when your phylogeny has the extant species or the existing species along the top, then the y-axis represents time. Okay, and that's a really important note because we hear sort of ill-conceived statements, right? Like, for example, humans are more evolved than bacteria. Oh, don't say that. Humans are perhaps more complex, but even then, humans, you also hear things like humans are better adapted.
That one bugs me.
That one bugs me too, right? And it's really important to recognize that humans have been evolving for the same amount of time as bacteria. Because they are all at the top of the tree. They are all extant species. And all the difference between humans and bacteria has to do with the branching and the relationships rather than anything related to how much time they have been evolving. We have all been evolving for the same amount of time. The degree of mutations and consequences is different. And that's wonderful. But we've all been evolving for the same amount of time.
It's yet another case where just like trying to identify the biases that you have, and in this case, a species bias that we have as humans is that we are somehow special is really hard to get rid of, even within evolutionary biology.
That's right. And there's this beautiful perspective in a book that we have on our reading list that we've recommended to read. This one here. It's the notion that, and I love this because it's a story, it's an idea that has been around for thousands of years before evolution, but the idea that humans are actually a new species, and we are. All of the evidence right now suggests that we've only been around for 200,000 years, maybe a little bit longer, but think about bacteria that have been around for like a lot longer. So the notion is that, humans are a relative newcomer to this planet, and we have a lot to learn from our ancestors or from nature, right? Who have been around a heck of a lot longer. Okay, so are we descended from monkeys? What do we think? What's the answer? Pop it in the chat.
I couldn't see the book.
You couldn't see the book. Here's the book. It's in the course outline.
And it is now in the chat as well.
Yeah. Okay.
Good.
Wonderful.
Nope, we aren't descended. We've just branched off.
Very good. Right? No, we're not descended from any modern monkey.
Yeah, that's the important one.
Or, yes, in that we are still monkeys. So it depends on your perspective and the way that you describe the relationships. That's what matters.
It's that context in relation to the tree.
Yeah. So not linear, definitely branching, and that has important implications.
Tree-based thinking. One last example with an article or a cover magazine from People, cover article from People magazine before you were born. In the early 90s, in the midst of the HIV/AIDS crisis and all of the rampant associated homophobia and demonizing that went on with that, there was this story that caught the media's attention of a group of people, 7 to 9 people in South Florida, who contracted the HIV virus and didn't know how they caught it. They, at the time, our understanding of the disease, they had none of the pre-existing conditions that were associated with people who came down with HIV. And so this was picked up in the media aggressively because, as I said, because of homophobia and kind of scare tactics associated with it. Underneath all of that, it was a bit of a scientific mystery as to how these people did contract this virus. And it was resolved actually relatively quickly using tree-based thinking. It was resolved in a way, this is the paper from Nature that did it. It's from Science, but you can click on this later on when you get the PDF and go and read the actual paper, download the sequences because they're all there. These are people, but this data is there and publicly available for you to look at. The story was that of the 9 people for whom this infection course was mysterious, they shared only one element in common, and that was a dentist. They had the same dentist, and they had all visited that dentist within the year prior to the onset of their symptoms. How are we going to resolve that? These scientists resolved it, these Center for Disease Control scientists in the early 1990s, which is right at the onset of being able for us to read directly DNA, to not indirectly look at it using allozymes, but to read the DNA sequences. They extracted DNA and they sequenced viral sequences from each of those people and compared them to the library of viral sequences that they had at that point, not yet. And this is the tree that appeared in that paper. This was published in the second largest journal in the world. And they used this tree to add evidence to support the hypothesis that the infection route was actually via that dental office. That was the hypothesis. And so if you're a nerd like me, you can go and download all of those sequences and I can present them to you in a tree. So this is the sequences of the patients and all of the known kind of Floridian diversity of HIV at the time, and I'm going to put a little star on to show you where the dentist was, and then ask a question. If that, and you're going to answer this with your stamps, if the HIV infection that the patients were exposed to was derived from the dentist office, where on the phylogeny would you expect to find the sequences from those infected patients? And you're all very well trained. So 1, 2, 3, go. When you're ready.
Thank you. Yeah.
Yeah, well done you.
Well done.
You're reading at the top level of the biggest top 2 journals in the world. And this was a really important thing. Now, how this happened is unclear, because this is like a really, I can't, sometimes you have to finish this description of the story. Yeah. It's a really sad story. Why, Dr. Smith? It's sad because all of the reasons you can think about, but one of them is that everyone's, except for 2 of the patients who survived to today, the dentist died very quickly in this story. And so nobody knows what happened. One of the symptoms associated with the onset of AIDS within an HIV infection is what do they call it? Neuropathy, where you lose sensations. So a working hypothesis is that there was a cut that he didn't know about and couldn't feel. Yeah. And so I've just drawn the box. We've cleared your very correct guesses. And so there, if I can, you see several clades in here. All the phylogenies have many clades, but this is kind of a conservative block, an inclusive block around where you would expect, in the diversity of Floridian HIV, where you would expect the dentist's patients to sit, if that's where the dentist sequence is, and if the infections had been derived from that office.
So that was a mutation tree, right, that was used in order to be able to understand the origin of a particular virus, a sequence. We've been doing lots of this in the media with COVID-19, same kind of thing. Yeah. It can be interesting, but it's also, when it's happening, it's tragic and has serious implications. And so that's one of the reasons why learning to read these trees, learning to make them, can be absolutely critical to supporting human health, right?
It's one of the things that we've noticed since the onset of this pandemic, is that this example, I mean, as you saw, that example is 30 to nearly 40 years old. Mm-hmm. And since 2020, we need less work to impress upon people why phylogenies affect your day-to-day life. Yep. How reading phylogenies affect your day-to-day life. I just have to reflect on the fact that I'm looking for a high five here, that I got through that and you didn't have to pick it up. Well done.
Okay, so here, some things for the midterm, for the topic test number 1. We're kind of wrapping up a few minutes early because we're going to imagine that you're going to have questions about topic test number 1. So pop them in the chat. Here's a thing that you might want to take with you as you study, some activities that you can do, kind of break down. Imagine that we have these 5 species of mussels. And some things you can do with your friends or that you can do by yourself to try to understand trees, to understand what they mean and what they don't mean. On Wednesday, we're going to do some 2.0 tree reading. So we'll test your knowledge on that as well, and hopefully plant some more flags for you for preparing for topic test number 1. We're also going to be next week opening up a asynchronous study tool, which is going to be a lot of work, and you're going to be like, oh, but the more that you contribute to that asynchronous study tool, the better you will do on the actual topic test. And so we don't want you to just go there and passively look at what everyone's done. You'll get more details about all of that. We'll tell you more details about the topic test on Wednesday as well, and make sure that you're all ready to go for it on October 6th, right? It was supposed to be the 7th, but we decided to make it the 6th so that you could have an extra long Thanksgiving weekend.
Somebody's noting in the chat that hopefully it's not cloudy tonight because Jupiter is going to be the closest point to the Earth, like for the rest of our lives, like even across our age difference. I think it's like 21, 29 or something the next time.
Yeah.
So, I mean, maybe you'll make it there. Probably not.
So, binoculars. You should be able to see some of the details of Jupiter with binoculars. Bananas. So, get outside tonight.
Now, to be clear, you can't see it with bananas.
Binoculars.
Binoculars.
Yeah. So, anyway. Any kind of question that you want, we'll be around. Otherwise, enjoy your day and hopefully rest up after the rather fun and long weekend that I think you all had. Okay, cool.
Well done, everybody.
Bye-bye.
We'll see you on Wednesday or in the chat in a second.